Good morning, class. Our today topic is plant breeding and hybridization. Plant breeding is also known as the green revolution, depend upon the green revolution, and to a large extent of the plant breeding techniques for the developing of the high yielding and disease resistant variety in case of the plant like wheat, rice, maize, etc. So, what is the plant breeding? Actually, plant breeding is the purposeful manipulation of the plant species in order to create the desired plant types and that are better suited for the cultivation, and it gives the better yielding and are the disease resist. So plant breeding is the technology to develop the disease resistant and high yielding variety of the plant. The main step in the breeding a new genetic variety of the crops are: number one is the step is known as the collection of the variability, means the collection and preservation of all the different wild varieties, species and relatives of the cultivated species. Second step is the evaluation and selection of the parents. Before going to make the hybrid crossing, we have to choose the selection of the very healthy and desired parent. So evaluation is done by the identification of the plant with the desirable character. The selected plants are multiplied and used in the process of the hybridization, means developing of the hybrid plant. In the third step, the cross hybridization among the selected parents. By crossing, hybridizing the two parents to produce the hybrid that are genetically combined, means the genomes are combined from the both parents and desired character is planted in a single plant in the F1 progeny. In the next step, we have to select and test the superior recombinant. When the recombinants are formed, we have to select and test it. The selection process is very crucial to the success of the breeding objective and require the careful scientific evaluation of the progeny. These are self-pollinated for several generations. We are going to, for the checking, for the selection, for the testing, we have to self-pollinate it for several generations till they reach a state of the uniformity, so that the characters will not segregate in the progeny. The method is known as the testing, and after the testing it have to release for the commercialization of the new cultivator. In the last step, this evolution is done by growing these plants in a research field, in a agriculture research field, in a botanical research field, and recording their performance under the ideal fertilizers application, irrigation and other crop management practices. It will be followed by the testing and the material in the farmer's field for at least three growing season at several locations in the country. So there are the three three main revolutions are found. One is the green revolution, means the crop production. Second is the white revolution, means milk production, and blue revolution means the fish production. So three revolutions are here: green revolution, white revolution and blue revolution. Wheat and rice. In 1963, several varieties such as Sonalika and Kalyan Sona is the variety of the wheat, which were very high yielding and disease resistant plant, were introduced in all over the wheat growing belt of the India. Semi-dwarf semi-dwarf rice variety were derived from the IR8, and Taichung's native one were introduced in 1966. Later, better yielding semi-dwarf varieties are the Ratna were developed in the India and the South India. In case of the sugarcane, Saccharum barberry is the name of that sugarcane, and Saccharum officinarum were crossed to get the desirable qualities of the high yielding thick stem. Both one is from the North India, another is from the South India. having high sugar and ability to grow in the sugarcane areas in the North India. So variety of the sugarcane is developed by the hybridization. Few millets crops like hybrid maize, jowar and bajra that have been developed in the India, which are very high yielding and resistant to the water stress. Because for the millet crops, it is going to grow to cultivation in the rainy season. So it have to resisted by the water stress or water logging. There is a requirement of this plant breeding for the disease resistant first, because method of this disease resistant for the various sequential steps, meaning the resist for the developing of the disease resistant variety. Screening germplasm for resistant. We have to screen the entire germplasm of the plant, germplasm in the total sum of the alleles, germplasm. The definition of the germplasm is that the total sum of the all alleles, all the different form of the genes present in the crop, and its related species also. So we have to screen all these germplasm. Hybridization of the selected parents. Now hybridization process is the most common method of creating the genetic variation. Hybridization is crossing of the two or more type of plants for bringing their traits together in the progeny. Third step is the selection and evolution of the hybrid. So parent is going to be selected is by picking of a seed and only those plant for the multiplication which have the desired trait. For example, the grain's length in the rice is variable, longer grain, intermediate grain and shorter grain in case of the paddy crop. Testing and release of the new variety. The newly selected lines are evaluated for their yield and other agronomical trait of the quality. So disease resistant etc. This evolution is done by the growing these into the research field and recording their performance under the ideal fertilizers, applications and irrigation. These are the crops. Wheat Himgiri is the variety. It is resistant to the leaf and strips rust disease, sometimes hill-burnt also. Brassica means mustard. Pusa swarin, it is the currant rye, white rust disease. So it is going to resisted with the white rust. Cauliflower, Pusa subra and Pusa snowballi. It is resisted from the black rot and the curl blight black rot disease. Cowpea means gram. Pusa komari, that is a bacterial blight. And in the chili crop, Pusa sadabahar, chili mosaic resisted from the chili mosaic virus, tobacco mosaic virus, and sometimes from the leaf curl disease. For example, in mung beans, in the mung dal, resistant to the yellow mosaic virus and the powdery mildew. Powdery mildew is a fungal disease. The powdery appearance found in the inflorescence were induced by the help of the mutation. And resistant in yellow mosaic virus in the bhindi is Avicenna muscus, and esculentus is the name of the bhindi, was transferred from a wild species and resulted in a new variety. This is called the Prabhakranti. Prabhakranti is the variety of the bhindi. Plant breeding is responsible for the developing resistant for the insect and pest also, because we now know that the insects and pests are very dangerous for the growing and developing crops also. So high aspartic acid sometimes and low nitrogen and sugar content in the maize, if it is there, it lead to the resistant to the maize stem borer. Stem borer can maybe cut the stems, because stems are the monocotic in nature. So if a single borer is going to be cut the stem of these maize plant, all the plants may be damaged. Smooth leaves and nectarless cotton varieties responsible to do not attack the bollworm are the worms that cause the disease in the cotton ball. So few crops like brassica, the rapeseed mustard, pusa gaurav, aphids controlled by the aphids insect, then flat beam, pusa same and pusa same 2 and 3, jassets, aphids and fruit borer. These are the insect and pests that may be controlled by these. And okra or the bhindi, Pusa swani and Pusa A4, the shoot and fruit disease, borer pest. In the case of plant breeding, it is also required for the improved food quality. So diet lacking essential micronutrients, particularly iron, sometimes vitamin A, iodine, zinc, it may increase the risk of the disease also. It reduces the lifespan and reduce the mental ability or retardedness also. So a far greater number, 3 billion people suffer from the micronutrients, proteins and vitamins deficiency, or sometimes hidden hunger. It is known as the hidden hunger because they cannot afford to buy enough fruits, vegetables, legumes, fish and meat due to the poor and the poverty. So more than 840 million peoples in the world do not have the adequate food to meet their daily food and the nutritional requirement. So we have to improve the quality by using this technology. Thank you and have a nice day.